Welcome everybody today to our GLIMPRINT seminar. Today is an off week, so I appreciate you coming to hear it. We have Nadeem Akhtar speaking today from Edinburgh on modeling of beta cell turnover in the pancreas. As always, I need to remind people that the meeting is live streamed and recorded and will be available in edited form on YouTube. Our seminar series here, we have information. You could use the QR codes to get additional information today. We are a group of organizations trying to support seminars and modeling in these kinds of areas. We hope that people will take advantage of that and participate in the other organizations as well. As always, Tom Helikar, myself, Jim Sluka, and Lorenzo are available to take your suggestions, critiques, comments, ideas. Please feel free to reach out to us if we can do things better. We have a variety of communication channels. They work better when you use them. Please help us get the word out about seminars when they're happening, and also about the archived seminar materials. If you have a publication to share, we can put that up on the IMAG page. We can also put it up on the GLIMPRINT page. The GLIMPRINT page also allows you to announce seminars, meetings, workshops, job openings, funding, and other things. If you have any trouble with those, please write to Jim Sluka, jsluka@iu.edu, and he'll help you out with that. Coming up, we have next week Shihao Yang from Georgia Institute of Technology. That's at a slightly different time. It's going to be at noon. November 20th, we have a talk from Columbia University. November 27th is Thanksgiving in the United States, so there's no meeting. We have Emily Luca coming on December 4th, and then Yom Berkut, who's already spoken once to us, coming back on December 11th. Please suggest people you'd like to hear. Suggest yourself, suggest your students, postdocs, anyone who is doing work in the broad area of computational modeling, which has an immune component, or experiments related to that, infrastructure, computational infrastructure. All of these things are appropriate topics for these seminars. Please suggest the people you would like to hear. If you've spoken before and you have new material to present, or you want to give us an update, don't be shy about suggesting yourself as a speaker. We will not have a steering committee meeting this week because we're an off week. Are there any other announcements before we move on? Okay, today's seminar is going to be mathematical modeling of beta cell turnover reveals non-trivial contributions of apoptosis to pancreas development. And I will turn the screen share over to our speaker.
Thank you very much for inviting me. So today I'll be talking about mathematical modeling of beta cell turnover during zebrafish pancreas development. Thank you very much, everybody, from my side, and welcome all of you to today's seminar. So I would just like to acknowledge before I start talking about my manuscript. So this work was carried out in the lab of Professor Nicola Nilo with strong collaboration with Dr. Lutz Bruch in the beautiful city of Dresden. Here is the manuscript. The link is here. So if you want to have a detailed look about the model and the science that we have done here, feel free to look up into this paper. So today, we'll start with a very basic introduction. So organ development, if you look at it, it's a very systematic process where we have different processes, cellular processes that work in tandem to bring about this organ development. We have the first process, differentiation, where you have these progenitor cells that subsequently mature to form a desired cell type. You have proliferation, where a cell can actually differentiate into proliferate into two daughter cells. So, and third process is apoptosis, where usually the dead or worn out cells are systematically eliminated from the tissue in order to induce tissue homeostasis. And this process of cell turnover, where you have this growth and elimination, it needs to take place in a very balanced manner in order to induce proper organ homeostasis and tissue function. Now, similar to any organs in the body, the beta cells, which are located in the pancreatic islet of Langerhans, also undergo this cellular turnover during early stages of pancreas development. Importantly, beta cell replication and differentiation has been well studied, but relatively less is known about the role of beta cell or the contribution of beta cell apoptosis towards the early stages of pancreas development. Now, this question is of fundamental importance because dysregulation in beta cell death is known to cause type 1 diabetes. And type 1 diabetes, this leads to lifelong dependence on a hormone called insulin. And we see that the incidence of type 1 diabetes is rising globally. Therefore, it makes it important to study the beta cell death and how beta cell death is dysregulated during this process of pancreas development and how it can lead to type 1 diabetes. So the important question that we wanted to address in this research was, what is the contribution of apoptosis towards the early stages of pancreatic islet development? And importantly, how does this beta cell death orchestrates islet immune system crosstalk? Because we know that type 1 diabetes is an autoimmune disease. So it could well be that during early stages of dysregulation in beta cell death could actually prime the immune cells to become autoimmune. So therefore, these are the two important questions that we wanted to address. So here we took an interdisciplinary approach. So we used zebrafish as a model organism, and we used genetic engineering coupled with mathematical model to understand the rate of cellular beta cell turnover during pancreas development. Now, historically, beta cell apoptosis has been very difficult to study. Why? Because as you can see here from this movie, in the green you have skin cells, and in magenta you have macrophages. So as you can see, that once the signal of apoptosis it takes place, the macrophages rapidly move to the target tissue and take up the apoptotic cells. Therefore, it becomes very difficult to histologically precisely quantify the amount of cell death that is taking place. So what we did was we took a counter approach where what we did was we inhibited genetically the apoptotic machinery. So we know that caspase is a protein that plays a vital role in inducing apoptosis. So what we did was we blocked the caspase using P35. So P35 is a baculovirus protein which blocks the activity of caspase. And what we did was we overexpressed this apoptotic inhibitor P35 under the insulin promoter, meaning specifically in the beta cells of zebrafish. So we are blocking the caspase specifically in the beta cells of zebrafish. And what we saw was that when we did longitudinal assessment of the beta cell mass in the wild type and the P35 mutant, what we observed was that so here in red we have the beta cells, and in blue we have the nucleoid, the DAPI. So as you can see that in the P35 mutant, we found that at 30 days, at juvenile stage, we found that there was a dramatic increase in the number of beta cells in the P35 mutant upon blockage of the P35, the caspase. So this made us wonder that, well, indeed, during this period of 15 to 30 days, we are seeing this increase in the beta cell number. And this increase could be because of the blockage of genetically blocking of beta cell death. So we wanted to mathematically understand what is the contribution of apoptosis towards this turnover. So previously in rodents, it has been done, the mathematical modeling has been done to study beta cell turnover. So there, in that study, what they did was they used a simple equation where they had replication and neogenesis as the factors that contributed to the growth of cells, and apoptosis that sort of led to this removal of apoptotic cells. And then they did this differential equation to understand what is the contribution. And as you can see, that they predicted, the mathematical model predicted that there's a wave of beta cell death happening at early stages of development, at early stages of pancreas remodeling in the rodent. But important thing what we need to understand here is that this article assumed that all the beta cells are equally proliferative, meaning that they did not assume the underlying heterogeneity among the pancreatic beta cells. We know that every cell type is heterogeneous, meaning that not all cells would proliferate at the same rate. And therefore, what we did was we expanded this mathematical model for our study. So basically what we did was we used these two open source software, Kupasi and Multicell. Here is a link to this website, which is open source. You can easy to use. You can access it. And so what we did was, first up, we had this experimental data. So we have this average on this panel, as you can see in the box here, we have average number of beta cells per islet in the wild type and P35 mutant, and we have the percentage of proliferating beta cells per islet in the wild type as well as P35 mutant. So we had this experimental data, and using this experimental data, we wanted to mathematically model what is the rate of contribution of beta cell turnover. So here we have NT, which is the total number of beta cells, and NP stands for proliferation and NQ stands for quiescence. So the total number of beta cells would be the sum of proliferation plus quiescent beta cells. So as you can see here that we are expanding the beta cell. We are making the mathematical model more robust as compared to the previous study because we are taking into account, first of all, two different experimental conditions. We have wild type and P35 mutant. And secondly, we are making this very clear distinction that the beta cells are not equal. We are incorporating heterogeneity in this study. So meaning that we have a specific set of beta cells that are proliferating and one that are quiescent. So as you can see, here is the differential equation, which gives us the sum of beta cell proliferation, and here we have the quiescent mass. So what we got was, so using this combinatorial approach, what we did was we tried to use this experimental data and see which model gives the best fit to the experimental data. So just to clarify here, first of all, so as you can see in the bracket here, the ones that are the parameters that are in gray are inactive, and the parameter that is in bold, that is active, right? And here you can see the orange dot corresponds to the beta cell, experimental beta cell number, and the blue one to experimental beta cell number in the P35 mutant. And the black solid line represents the model of the wild type. And the orange dotted line represents the mathematical model of the P35 mutant. So as you can see that if we have only proliferation that is going on in the mathematical model, that model would give an exponential increase in the beta cell mass. And it provides a very worst fit to the mathematical model. As you can see with the root mean square, it's very high, meaning that the mathematical model is not fitting well with the experimental data. So what we did was we gradually started to add more and more experimental parameters to the mathematical model. And as you can see from here, that when we incorporate neogenesis, proliferation, as well as apoptosis, you can see that the model starts to align slightly better. Similarly here, as you can see that, but when we don't have the quiescence, for example, in the second graph, I just want to point out here, when we don't have quiescence, we still get this exponential increase. But as soon as we incorporate quiescence, we see that the model starts to fit very nicely with the experimental data. Also, you can see the root mean square is quite low. But here, one important caveat is that we still don't have the apoptosis part. But on the graph here, as you can see, when we incorporate all the parameters, meaning you have proliferation as well as apoptosis and quiescence as well, which is important. So beta cells can actually switch between proliferation and quiescent state. We see that the mathematical model provides an excellent fit to our experimental data. That is the take-home message that we found. And the important thing here was that the mathematical model made an assessment or made a prediction that the apoptosis rate was 2.25% per day. That was the important take-home message that we found from the mathematical model. So now the question that we wanted to address was that can we corroborate this finding experimentally? So to this end, we did tunnel assay to histologically quantify beta cell apoptosis. So tunnel assay basically helps us to label fragmented nuclei. So it's a marker of apoptosis. So as you can see here, the beta cells are in red, the tunnel positive cells in green, and the DAPI in gray. So as you can see that we found very sparse rate of beta cell apoptosis in the islet. So meaning that histologically we could not corroborate the mathematical model of 2.5% of beta cell apoptosis. So the obvious question was that why is this discrepancy? So the one important parameter, as I discussed before, was the macrophages. the immune cells that play a very important role in quickly coming and engulfing the apoptotic cells. And then what we did was we wanted to study, well, is it indeed that the macrophages are coming in and taking up the dying beta cells? That's why we are not able to confirm these, corroborate the experimentally the beta cell apoptosis. And lo and behold, that was true. Because as you can see here, the beta cells are in green and the macrophages in red. So as you can see that at 20 days already, there is a significant infiltration of macrophages in and around the islet. This would mean that indeed the macrophages are coming in, taking up the apoptotic beta cells. That's why we are not able to see it. So we asked a counter question. Well, if we block the macrophages, can we see evidence of apoptotic beta cells? So to this end, what we did was we used a mutant zebrafish. So we had a IRF8 mutant, which is basically IRF8 is a transcription factor that plays a role in macrophage maturation. And as you can see that in the here the macrophages are in red. So in the wild type, we have normal distribution of macrophages, but in the mutant, we have no macrophages at all. So we used the wild type and the mutant fish where we had no macrophages, and we did a similar assessment. So green beta cells and tunnel in magenta. So as you can see that in the mutant cells, mutant zebrafish, we saw an increased number of apoptotic beta cells debris in the islet, which kind of fits very nicely to our model now. So the model makes an assumption, provides an estimate. We are not able to corroborate histologically because of the presence of macrophages. And only when we deplete the macrophages, we are able to see the evidence of apoptotic beta cells. So now we wanted to make the mathematical model much more robust. So previously, as I showed you that in the P35 mutant and the wild type, we found that there was an increase in the beta cell number between 15 and 30 days, right? So the question that we wanted to ask was that when exactly is this increase taking place between this time window? So what we did was we added a lot more experimental parameters now. So we had 20 days, 23 days, and 27 days between the wild type and mutant. And what we observed was that as early as 20 days, we are seeing that there is an increase in the number of beta cells in the P35 mutant where we block the beta cell death, meaning that the beta cell death could actually start as early as 20 days or before that, because that's exactly where we see the increase. So we also incorporated this experimental data into the mathematical model in order to make the model much more robust. So here, as you can see, so we had the similar mathematical model, and here the model makes an assumption that the beta cell apoptosis starts at 19.5 days. That's the onset of beta cell apoptosis. And here the model says that, well, what if the wave of beta cell apoptosis, as we've seen before in the rodent model, where we had this sudden peak of beta cell death and it subsides subsequently. So what we did was we used that same strategy, mathematically modeling, to make a wave of beta cell death and see how it fits. So as you can see that if we make a wave of beta cell death, so just for one day, we see that the model does not fit very well to the experimental data. But then what we did was we made a duration, meaning that apoptosis starts at around 17, 16 days, and then this apoptosis duration continues all the way till 30, 35 days, till the experimental data we achieved. So meaning that we had this gradual onset of beta cell death. And this model actually provides a very nice fit to the experimental data. So that's the key message here that we wanted to show, that unlike the rodent, in the zebrafish model, we have an onset of beta cell death that continues, that starts early in juvenile stage and that continues until late stage of pancreas development. So that was a very important finding that we had, which is not similar to the rodents. Right. So that was about the ordinary differential equation to study this process. So we also wondered, so we have the model where we are using differential equation to study this turnover. We also wanted to study if we are able to sort of study this process in more stochastic manner. So we have like a microscopy image where we had this growth of pancreas can we make more like a stochastic model where we can study this turnover of beta cells so as to mirror what we are seeing histologically? So we use another approach where basically we use this open source software, which is Morpheus. The link is here. All of us can access it. And what we did was we used this a stochastic or distribution, a random distribution of beta cells to study what is the turnover. So as you can see here, we have the wild type cells, P35, and here we have the time window. The red cells are quiescence, the green cells are proliferation, and the blue ones are the apoptotic cells. So as you can see very nicely, that even this we can use a different model approach where we can use more like a stochastic distribution using this Morpheus, and that also provides us with a very nice readout of this cellular turnover process that is happening in the course of beta cell development. And this process actually is very cool because it gives us a picture of how we could microscopically image the beta cells. So a similar image that we can actually acquire using this mathematical modeling tool called Morpheus. So just to wind up, that was about the paper. So the story does not end here. Actually, we would like to continue with this study that we have found even in the future. So, for example, in the previous model where we showed that cell turnover taking place, we have quiescence, apoptotic cells, and so on. So we can also incorporate in the Morpheus model the immune cells, because we know that if there's apoptotic blue cells that are coming up, apoptotic cells, the immune cells will come and pick it up. So we want to expand that model. And currently, I'm doing a postdoc in the University of Edinburgh, and here we are looking at basically skin cells again in zebrafish, uh, studying oncogene and tumor expression. And we know that in tumoric conditions, the skin cells proliferate rapidly. And what we also found here. Sorry. So basically, I want to show here is that so in the green we have the skin cells, and in red we have the neutrophils. And when this tumor cells expands and there's turnover of the tumor cells, the neutrophils are rapidly coming in as well and playing a role in this cell turnover process. So we can actually incorporate this process as well and expand the Morpheus model that we have previously. So we would like to take this into account as well for the future study. So yeah, so thank you very much for your attention, and the session is open for discussion. So we can just to wind up, I would like to just say that you can just click here. This will directly take you to my paper, and so when you go to the paper, you basically just need to click about on the data availability, and once you come here, you just need to click on the Morpheus model as I explained to you before, and this will directly take you to the website. So here, as you can see, we have the nice explanation about the beta cell turnover in the pancreas. And this gives a very nice explanation. As I explained to you before, this is the movie that you can play it again here. And you just need to click in the green box here. And this will directly take us to the Morpheus website. So this is exactly what I wanted to wind up with. So you just basically need to click on the link of the paper and data availability, and it will directly take you to the mathematical model. So everybody can have a look. It's open source, and and you just need to click on different parameters just to check, what you want to see how this was done, and it's very user friendly. You have the nice description here on the right hand side, for example. So everybody can take a look, and if you just click on the run here, for example, it runs automatically. So meaning the stochastic distribution that we saw previously, everything will come up here. And for example, you can click on the different time points, and this will show you the cell numbers during this course of development. So yeah, it's pretty easy to use, and everybody can can feel free to use it. So thank you very much for your attention, and if you have any questions and queries, we can talk about it now.
Great. The floor is open for questions. I'll start out by saying this is, of course, a very impressive example of making software available for use and potentially reuse. What do you see as the biggest challenges that you had in terms of presenting the software and making it available in a user-friendly way that you've described here?
We didn't really face much of a challenge, I would say, because so we had this open source software that we basically used to mathematically model this process, and it's free to access. So pretty much everybody can access it, and it's easy to use the graphical user interface.
And if we were going to try to adapt or reuse this model,
Yeah.
what would the support be for doing that, and what would the challenges be?
So basically, we can actually, so the model is quite user-friendly, meaning that, we can incorporate more and more parameters into the mathematical model to make it more robust. As I said, for example, we can incorporate the immune cells and see how they are dynamically coming in and picking up the macrophage, apoptotic cells, for example. So we can make the model more and more robust, I would say. So that's the advantage of this model. And in terms of challenges, um, I would say that perhaps we would maybe need more experimental data, more and more experimental data, to back it up. So maybe the model we can start off perhaps by using simplistic approach, simple experimental data, but then perhaps we might need a lot more experimental data to make it more comprehensive or more conclusive, so to speak.
Thank you for this very interdisciplinary work that you actually do, combining experiments, wet lab, and then also modeling in various frameworks, and probably using all the strengths of the different approaches. So coming back to the first introductory slide where you discussed the contributions of proliferation and apoptosis to organ development, and you said there is this delicate balance of proliferation and apoptosis. And the model as it is now, it assumes that there is some constant rate or probability of individual cells to do proliferation and to do apoptosis. And it seems that evolution may have tuned these numbers well so that the islet of Langerhans has the proper size and then can function well in glucose control. But that doesn't sound robust. If there's a perturbation, the cells should respond. And at the same time, it also seems wasteful that all the organs would first proliferate too much and then through apoptosis reduce to the correct islet size. So probably that hints to some deeper regulatory processes that we miss so far in the model. And it's of course now a challenge for new experiments, maybe deleting some factors that could control these processes. So what are possible inputs into the regulation of both the proliferation part and the apoptosis part that would feed back basically the state of the whole islet? One could imagine that the cells communicate among each other to get a sense of how big the islet is, how many beta cells are already there, and that could maybe inform the individual cells whether they need more proliferation or more apoptosis. And then this could all be feedback regulated and then become a really a robust organ size control mechanism. And for that, you need apoptosis, because at some point, if you had very good microenvironment, all the cells were very happy, proliferating well, then the islet would become too big. So you need apoptosis, but maybe not all the time, only in those cases where the conditions were so good that you just would end up with too many cells in the end. So what could be biological control mechanisms that one would need to add into the model that currently has basically individual cell parameters that are constant and not controlled?
Right. More like an intercellular communication, so as to make the islet size more controlled. Yeah, I agree. So the intercellular communication can be added, perhaps, how the cells are communicating with each other in order to enforce this proper islet size.
And are there already candidates? Do you know of molecules? How would beta cells communicate?
Right.
To
So for
see how
example,
many cells are there?
yeah, right. So for example, if a cell is dying, the apoptotic cells would release a molecule called ATP, adenosine triphosphate. And there's already experimental data that have shown that we can tag this ATP molecule with a fluorophore. And these ATP molecules that are being released would signal. So we have the apoptotic induced proliferation. So the cells that are undergoing apoptosis release these factors that potentiates other cells to proliferate as a compensatory mechanism, for example. So these molecules, for example, ATP, and then we have these gap junctions called pannexins. So these ATP molecules that are being released take up by the neighboring cells via these gap junctions, and they communicate. Apoptotic cells and the neighboring cells communicate via this process. So in future, perhaps, we could use this tool to really to tag these cells that are undergoing apoptosis and then see if the neighboring cells starts to proliferate based on the cues that are coming out from the proliferating cells.
Very nice. So I think this is a good basement for new students probably coming in, making the same model that you have just presented, perturbing the number of cells by artificially deleting some cells and seeing that the current model cannot recover from such damage. But if you include exactly what you said, a signaling mechanism that other cells sense, oh, there was damage somewhere or something like this, that they could respond, and then one would get a more controlled organ size, which probably is what physiologically is much more needed and probably exists.
Relevant.
So a very good starting point. Thank you.
Lorenzo.
If I look at the images, the very nice image that is in your synopsis of your paper, the experimental one, I see your macrophages and your beta cells, and I wouldn't be surprised at all if those macrophages were perivascular, because I can almost see the blood vessel, honestly. Well, that's my job, so I'm biased, but yeah, my question is, I wonder if you have considered the microvascular involvement, or you are thinking to consider that in the future.
Right. I think it's an excellent question. So we actually did some imaging using the FLIC GFP. So FLI is the transgenic reporter line, which actually labels the vasculature in zebrafish. And actually, we found that the zebrafish islet is quite densely vascularized. And we find that many of the macrophages are actually sitting near the vasculature of the pancreatic islet. But actually, I didn't really follow it up that extensively as to how dynamic is this process. Whether the important thing is that whether the macrophages are coming out of the vasculature and staying in the islet, or are they going back and forth, meaning that they are mobile. So because we know that we have this different type of immune cells. One are the tissue resident immune cells, and the other ones are more like a sort of coming in, going out, based on the different pathological conditions. So based on my experimental experience, I would say I did a couple of times this experiment and just found that there were some macrophages that are near the vasculature, but I didn't really do a robust assessment to see how many of these macrophages are tissue resident and how many of them are coming from the vasculature and going back. So yeah, that would be an important question to tackle. I agree with you, Lorenzo.
Thank you very much. Also, the direct signaling of the endothelial cell to the pancreas may have, so it can be a three-way crosstalk. But thank you very much. That was very nice. Thanks.
So the question was, what is the underlying model behind Morpheus? How does Morpheus work?
So Morpheus basically uses Kupasi as a model. And so, yeah, and then we have these different cellular parameters that we incorporate. So we have different, the ordinary differential equations, and so different cell types. So we incorporate all these things into this Kupasi, and that's how we use this Morpheus to run the model.
I think perhaps the question was more, how do you do the multicellular model? How does the agent-based component of it work? And then there's a follow-on question, how would you add more cell types to this model?
Yeah, for example, if we come to the left panel here, for example, here cell types. So we can, as you can see, that we already have all these different cell types here, for example, the P35, the wild type, and so on. So we have the chance of adding the different cell types, adding more parameters into this function, so to speak, in the Morpheus.
Regarding the spatial layout, I mean, this is a cellular POTS model, but at the moment, it doesn't make use of all the nice mechanical properties that the explicit cell shape provides in the cellular POTS model. It's basically illustrating the spatial distribution of the cells, giving them a shape that is generated by the cellular pods model. But all the processes that go on here, proliferation, quiescence, activation, and so on, are independent of where the cell is sitting and what its neighborhoods and so on are. So the CPM is mainly illustrating how such an arrangement of cells would look like and how the apoptotic cells would be distributed, and that may guide then how you set up your experimental microscopy focus and so on, if you have an impression of this in space.
So this doesn't include signaling then between the cells?
Not yet.
No,
Okay.
not yet.
Okay. So Idamar asks, can you consider metastasis and the cell signaling microenvironment if you were going to expand this model?
Yeah, we could, as we discussed before. So, for example, the signaling environment, for example, from apoptotic cells, for example, if there's any cue that's coming up, so we can actually spatially look at the distribution of the apoptotic cells, for example, and how does this apoptotic cells influence the neighboring cells. So, meaning that would give us a readout how this signaling cues emanating from the apoptotic cells can actually influence the neighboring microenvironment, for example. So we can actually make this model a little more robust, I would say.
What would be the biggest challenges to including cell signaling in this model? What would you have to do to extend it to include cell signaling?
So first of all, I think we have to potentially label the apoptotic cells, for example, the one that are sending the signal, and then potentially we have to have the cells that are receiving the signal. So for instance, I would say like we have the ATP fluorescence dye to label the cells that are releasing these things, and the cells that are receiving it, for example, we can tag them with a gap junction protein, pannexin or whatever different with the fluorescence. And then potentially it would be that, okay, we can label these cells in the three-dimensional or two-dimensional space, and then see how does that influence the cellular turnover process, for example.
And so Idamar asks a follow-up question, which is, how would cell signaling and apoptosis potentially block metastasis, and how would you potentially put that into a model? Oh, I think the question was more on metastasis. If you're thinking about this as a way of regulating the tissue so that it's going to avoid the development of initially probably benign hyperplasia, then later the potential progression to cancer, how might that work?
Right. I mean, it's an excellent question. To be honest, in terms of metastasis, as we know that the cells would migrate to a distant organ. So the point would be that, how many of the cells have the potential or tendency to migrate from the target from the host environment to the target tissue. So I think the question would, so then we would have to look at the underlying heterogeneity, or which are the cell types that can actually progress. And if these are the ones that are progressing, for example, then we have to look at the molecular signature of these cell types, and then potentially try to block it, and then see if indeed that is preventing the cells from metastasizing to the distant organ.
So I know one thing in pancreas, when you try to do chemotherapy or radiation, is that because the pancreatic cells are producing rather reactive molecules, they have high ROS levels to begin with. They have very high levels, for example, of glutathione, and that makes them quite resistant to killing by cytotoxic chemotherapeutics or by radiation. And so that's one of the reasons they're quite difficult to treat with standard therapies. And apparently it also makes them resistant to immune killing by T cells, for example. What would it take perhaps to include that kind of essentially metabolic or reactive oxygen species buffering? It's not necessarily going to be the apoptotic pathway that's turned on. It might be a pyroptotic pathway or another kind of cell death. But what would it take perhaps to include those other kinds of cell death? and think about what makes these specifically pancreatic cells rather than other cell types.
It's a good point that we have different types of cell death, not only apoptosis but necroptosis, ferroptosis, curoptosis as well. Yeah, I think it would be important for us to really understand that under which condition, what is the cell type, what is the mechanism of cell death under different conditions. So, for example, here we took into account apoptosis because we knew that during this developmental process we had this apoptosis process that actually contributes not much of rather other ferroptosis or things like that. So that's the reason why we took into account this process. But of course, necroptosis, for example, could actually contribute as well. So we know that in necroptosis, we have the RIP3 kinase, the receptor interacting protein kinase, MLKL3. So there are different pathways that contribute to this the necroptotic cell death. So it would be important for us to sort of experimentally first understand that in tumorigenic condition and in normal developmental condition, what is the mechanism of cell death. And once we identify that, I think experimentally, then we could potentially try to sort of take it to the next level and try to understand that in pancreatic cancer or things like that, what is the relative contribution of these different cell types and how they are undergoing these forms of cell death, for example.
You mentioned how you were looking at using tunnel, for example, to look at apoptosis. What would the challenges be in terms of looking for other cell death mechanisms in your current experiments?
I think the first important thing would be that if we sort of take into account that if the cells are dying via, for example, necroptosis, let's assume that one. So the first important parameter to consider here would be that the mechanism of cell death in necroptosis is slightly different, as I explained, the RIP kinase and the MLKL participating in this form of cell death. So the idea would be that to target this specific pathway, perhaps, and then see if we can target this pathway and block the cell death. That's the one way. For example, targeting RIP3 kinase or MLKL. The second possibility would be that normally we know that apoptosis does not contribute largely to inflammation. And the cells are sort of taken up. They form small debris and they are taken up by the macrophages. But in terms of necroptosis, we know that they're highly, potentially they might actually lead to inflammation because they release the intracellular contents. So one way would be that to check the inflammatory profile of the islet and see that it is excessive, inflammation, for example, and it's mostly coming, and this excessive inflammation is mostly coming from these cells that are dying. Then that would mean that yes, indeed, these cells are undergoing necroptotic cell death, and that is contributing to the inflammation of the islet, and therefore, we can have this another parameter added to the model, for example, to segregate apoptosis with necroptosis, for example.
So Idamar says it would be interesting to have markers that allow you to monitor cell cycle and differentiation fates.
I just forgot to mention one point with regards to the cell cycle, actually. So we do have in the pancreatic beta cells of zebrafish, we do have the cell cycle duration, which normally takes around 24 hours. So we took into account the duration of cell cycle for 24 hours. But yeah, I think with regards to differentiation, I think we can, sort of try to take into account that part as well. Yeah.
So one other possible question for me would be, you're switching your organ system, but you've mentioned that a lot of the technologies you developed when you were at Dresden are carrying over to your current work. Can you talk a little bit more about how you've used the methods and approaches that you developed in Dresden now for your current project on skin analysis?
So first up, what we are actually looking here is, again, tumor initiation. So like at early stages of tumor induction, not really at late stages, because we know that early stages we have this remarkable turnover of the cells, and how does this cellular turnover actually influences the immune cells. So for to study this, what we have done right now is that we are developing a lot of tools. So for example, the first one we have is the live imaging of apoptotic cells. So what we have done is we have made a zebrafish transgenic line where we had this annexin YFP. So we had this annexin, which is basically a membrane tethered molecule, and this annexin is tagged with YFP. So when a cell is undergoing apoptosis, the membrane disintegrates. So it flips, not really disintegrate, but the lipid bilayer flips, and we have this bright YFP expression. So with this, we can actually in vivo visualize the number of apoptotic cells at a given time frame. So we can have, let's say, cancer cells labeled in green, for example, with GFP, apoptotic cells with YFP, for example, labeled in magenta or something, and then we can have the neutrophils and macrophages labeled with different colors. So what we can actually do is we can incorporate more and more variables into this mathematical model and say that, okay, we have live documentation of so many cells that are undergoing death, and that leads to the recruitment of, so X number of cells that are dying leads to the recruitment of Y number of immune cells. And this is the process that is sort of orchestrating the crosstalk between the two processes. So right now, I'm in the process of, developing a lot more in vivo tools to visualize these processes of cellular turnover in the zebrafish skin, so we can make the model more robust. So we are making the cell death reporter marker. We are making the Fuji, which is again a cell cycle reporter. So we are making the Fuji a marker, a transgenic marker, so we can label the proliferation and the quiescent cells. So we can have the proliferation cells in green and quiescent cell in red. So we can actually segregate these population of cells in real time in a longitudinal assessment. For example, we can say that, well, we did this oncogene induction in zebrafish at 3 days, for example, and we followed the skin cells from 3 days till 7 days, for example. And then we can microscopically say that we analyze so many cells that are proliferating, so many quiescent, and so many apoptotic. So right now I'm in the process of developing this in vivo tools that I also was working with in Dresden, so we can make the model more robust and see what we can take it up from here in terms of the cancer research.
Okay, well, I want to thank you very much for coming and speaking to us about this nice work, and I hope we can maybe have you back and talk about the work you're starting now
Yeah.
and developing in the future.
Thank you very much for having me. Thanks all of you for coming. Thank you.